Welcome to Virology. So, we gotta start somewhere, and it might as well be here. What's a virus? What is a virus? The answer has evolved over time, and there isn't any one answer that you can get all the virologists to agree on. We're getting close, but there's still massive controversy in this area. So, here is how we got here. We start out with the virus as a concept of some kind of poisonous substance. Originally, people think it's probably either something you get through the air, like a smell, or something that's a liquid. Now, the people were getting sick with viruses long before we had electron microscopes and molecular biology, and so at first, a virus is kind of an idea. It's this bad thing that happens. And mostly, viruses were defined by what they did, by making you feel sick, making you feel bad. And we've moved on since then because, what happens if you get a virus that doesn't make you sick? Yeah. But to show you how new this entire field is and how little we know, let's go through a little history. Yeah, we still thought it was actually called a contagious living fluid. 1899, that was the leading scientific conjecture. Then we thought this thing's made of protein, and then we eventually figure out it's actually got some nucleic acid. The first one they looked at happened to have RNA inside, but later we found that most viruses have DNA inside. But that's good, because I like RNA viruses. I think they're the best. Then we learn about the shape of it. We learn that it's really tiny. And so we got a thing with a shape, but we don't get the first virus sequence until 1976. And so think about that. Your parents may be older than the first virus sequence arriving to humanity. And so what that means is that we have a hard time making long-term predictions about what viruses can or can't do, what they will do, how they'll change, how they won't change. You'll see people out there making these predictions with a lot of confidence, but it's confidence built on, you could say, hopes and dreams, or you could say nothing. And I think either of those would mean roughly the same in this case. I come from the field of coronavirology. I've studied coronaviruses almost since day one. Yeah, since back in the 90s. Yeah, how's that for a dunk? First coronavirus sequence actually came out before I started in the field. 87, I was in elementary school or something. And then we gradually figure out about virus evolution. And we have original SARS in 2003 and new improved SARS causing the disease COVID-19 just a couple of years ago. So, what's a virus? It's easier to tell you what most viruses are than to lay out a sentence that says what a virus is, because viruses are so weird that there is always some virus out there that breaks anything that you try to say, that makes any true statement ever so slightly false in one case. They are weirder, more variable than any of the branches of life. And that sometimes makes them a little bit difficult to study because a lot of the tools that are made for bioinformatics, for example, work really well on living things like cells, and they just break and quit and cry when you get to viruses. That's just how it is. A virus normally encodes at least two things, one of which is a thing called a polymerase, which is going to be a protein that copies the virus genome. So it's going to have a genome, and that genome can be RNA or DNA, but we'll show you that later. And the genome is going to encode two proteins, one of which is probably going to be a polymerase, one of which is probably going to be what they call a packaging gene, like a thing to wrap up the genome. And it can wrap up the polymerase and the genome, or just the genome, depending on how it wants to do business. And so there are tiny viruses, and that's all they are, just those two parts. And there are giant viruses that have those two parts and a bunch of other stuff. And of course, there are exceptions. There are viruses that kill people every day that are lacking a polymerase. And we'll go through those later on. There are plenty of viruses in the world that lack their own package, basically a thing that makes a little virus particle like this awesome, totally inaccurate, but still nonetheless awesome animated GIF. So let's go. So this is a virus. Now, when the virus is outside the cell, your first vocabulary word is virion. And a virion is also called a virus particle. It's a little ball, and on the inside is going to be the virus genome, and on the outside is going to be some kind of protein that is going to help this virion find its way into a cell. And so probably that protein is going to attach to the cell, just grab it, and probably it is going to have some way of essentially punching a hole in the cell and carefully depositing the virus on the inside, the virus genome. Once the virus is in the cell, it doesn't look like this. This guy is what people think a virus is. When the virus looks like a little spiky ball or like a little lunar lander, that's when it's the virion, and the virion is metabolically inactive. It is essentially the dead form of the virus. When a virus is alive, it looks a little more like this. Look at that. So this is a network of interconnected chambers. You can see how they're all hooked up. And these are created by a chicken coronavirus as it is growing. So this is the replication factory that the virus builds. It only exists in virus-infected cells. And it is created with virus proteins. And you can just put in the correct virus proteins in the right amounts, and they will do this to a cell. So it's a thing that the cell does in response to the virus, and a thing that the virus causes the cell to do for the benefit of the virus. There's no benefit to the host cell from doing this. This is essentially the virus playing puppeteer with the cell. Yeah. So that's neat. Yeah, we'll play this in class. And all right, so let's round this out. So what's a virus? How do we define it? At the top, I've put what I would consider to be a pretty bad definition, but this is like if you asked a person on the street, they probably wouldn't give you much better than this, and it's not the worst. Yeah. So microorganisms smaller than the bacterium that cannot grow or reproduce apart from a living cell. Now, in this course, we'll show you that all the statements in red are wrong, and all the statements that are not in red are not actually statements, they are connecting words. So every part of this is wrong in some way, but most of the parts are generally correct. So a virus is not really a microorganism. It's not actually alive in the sense that things that we call living are alive, although that is a point for debate. And yeah, I think the only problem with calling viruses alive is that you have to change the definition of what alive means. And I think people are a little squeamish about that and the implications of that. So yeah. But whatever, there are reasons for things. They're just not always sensible reasons. There are giant viruses, and some of these things are bigger than the smallest bacteria. So that is false, but only sometimes. Usually, viruses are smaller than the kind of cell that they grow in, and the giant viruses actually grow in giant cells. So yeah, they're smaller than their host, but they're not smaller than every cell. And viruses cannot grow or reproduce apart from a living cell. Well, there was a really cool experiment where they did exactly that, and there have been several since where they do exactly that. It's not that they can't, it's just that they almost always only replicate inside of cells. But if you have all the components from the inside of a cell, even if they're not inside of a cell anymore, that's good enough. The virus doesn't have any way to check, its environment, look around and say, hey, this is weird. Yeah, I don't remember being in a test tube. Okay, so bad definition, but generally it's got some merit. It's not that it's wrong. You can't really say anything wrong about viruses because you can't really say anything right about viruses. They're not really any universals that you can hold on to. They're just ideas. And so, yeah, one idea is that we have mobile genetic elements. So break that down. That's the most important part of the current virus definition, which, fast forward a couple of years, will have changed, I guarantee it. Um, but mobile means it can move from cell to cell. And the cells are both still there at the end, potentially. They may die very soon after, but both the components of the cell remain in place. But this little component goes out, and it goes from one cell to another, like a little bee pollinating flowers, except it doesn't pollinate, it infects, and they usually die. Yeah. So it can copy itself, usually, but there are exceptions. There are viruses that have no polymerase. There are things that we classify as if they were viruses that have no proteins whatsoever. So we'll get to those. And they grow in the context of the inside of a cell. That's better, because that's saying they grow in the stuff you would find inside of cells, which is actually a true-ish statement. You can probably find an exception to that, but I don't know. I think that's reasonable. The current definition from the International Committee on Taxonomy, which is like naming and organizing of viruses, says that they're mobile genetic elements, so yep, I like that encode at least one protein, or, and get this caveat on the end, or they're actually descendants of something that used to encode one protein. So you've got to show that they can at least make one protein, and that, or that they are genetically similar enough to other viruses that do make one protein to say that, yeah, these are part of the same lineage, they're part of the same family line. And so if you're descended from a virus, that makes you a virus by default. That's the idea. Yeah. The other part of this, and this is the part that I think may have to go away sooner rather than later, is that at least that one protein that they encode, they can encode more, but they have to include at least one, is a component of the virion, like a protein that makes a virion. Now, as soon as we've made that, we actually go into the exceptions. And right underneath that in the ICTV code, there's this paragraph of exceptions. Long terminal repeat retrotransposons. So those are things like HIV that are loaded into your genome already. Yeah, you've got a lot of these things in you. And not because you're infected, but because our ancestors back through different kinds of cavemen, if you want to call them that, were also infected. And so we have the same viruses at the same spot. Phages. So these are viruses of bacteria. They used to be treated as if they're somehow different. And I think part of that was just thinking that humans are really special and we couldn't possibly be taken down by the kind of thing that would take down E. coli. But here we are, and they definitely do. Paulintons. Yeah. These are a different class of virus-like transposons. So a transposon is a piece of DNA that has the ability to make a protein, usually, that will cut out that DNA and then can move it and find another chromosome, open it up, and glue in the DNA. So it's a little piece of DNA that's actually capable of basically crawling around in your chromosomes. And sometimes, while it's crawling, it'll actually get duplicated. It's 1 out of every several hundred human births actually results in the duplication of one of these crazy little transposons. Yeah. So, yeah, that's weird. It creates some unpredictability, and there's nothing good about having them there in particular. It's just that we are infested and have no good way to get them out short of wholesale genetic modification of all human beings. And I don't know that humanity is quite ready for that yet. I also don't know that the technology is quite up to snuff. Okay, so those are all exceptions. And satellite viruses and virophages. So that's 2 classes of things that are able to infect virus-infected cells. So a virus just needs a host cell to go into, but virophages and satellite viruses are kind of names for the same thing. They have to go into a cell that's already been infected by a particular virus. and then they can actually be copied or copy themselves. They're going to steal some components from the host virus and the host cell. So literally virus of a virus. Yeah, dirty little things. And then there are capsidless dependents. So capsidless, a capsid is named for a protein that makes a shell around the outside of a virion. So this is supposed to be a coronavirus. It's not really very coronavirus-y, but it's trying, and it's cool, and it pulsates, so that's why it's there. Capsids are like for viruses that have a hard protein shell around the outside, and that's just the name we give to their protein. And some viruses that are related to viruses that have capsids have lost their capsid genes and still somehow find a way to move around and be mobile genetic elements. And so all of this stuff at the front is listing exceptions, and it says, yeah, we're going to consider these to be viruses sensu stricto, in the strictest sense. Yeah. So they are literally viruses, even though they totally violate some of the rules of paragraph subsection above. Yeah. That's the problem you run into with virology. Just mayhem, chaos everywhere. And then to top it off, you got these other things. And this is the paragraph underneath the paragraph of exceptions. So there are a bunch of things that we're going to pretend are viruses, even though they're not viruses, but we are going to classify them along with the viruses in the same sort of way as if they were really viruses. Yeah. So like Pinocchio viruses, we'll classify them as if they were real boys. Oh my gosh. So two of these groups are called viroids. And viroids are neat. I'll show you a picture here in a sec. But they are uncoated, so they have no capsid, circular, single-stranded RNAs, or things related to them and descended from them, that sometimes have these things that are called ribozymes. It's like an RNA that folds up into a shape that does a job. It's an enzyme that's just a string of RNA. Its sequence determines what properties the enzyme has. It's crazy stuff, honestly. But yeah, viroids have them pretty commonly. And there are these things called polydnaviruses, which you can break that down. Poly, and like, oh, okay, several. DNA, and virus. Yeah, we know virus. Yeah, we don't know what it means, but we've heard the word. If you haven't heard the word virus by now, you're doing great. Wow. I don't know what COVID was for you, but good job. Yeah, way to look on the sunny side. So these are neat. They are most common in parasitoid wasps, so ones that will lay their eggs into caterpillars of butterflies and stuff. And then these are genetic elements that will come from the wasp. They will be transmitted alongside the baby. They will grow in the baby. They'll also grow in the host caterpillar, although they just seem to go into those cells. They don't seem to come back out. And they can be transmitted in a virus-like way. So they're embedding themselves in the genomes of the babies and are then transmitted on. Yeah. So, and it's not the sort of thing where you can say, well, why does nature need that? Nature never needs viruses. Viruses need nature to replicate in. Yeah. We're here for their amusement, I guess. So, yeah, viruses are strange. All right, that's enough for this section. Thank you very much. I hope that, was interesting or blew your mind or whatever. And we got more weird stuff around the corner. Check out the next video. Thanks.